Jerry, for your generous to be here and to have the opportunity to speak to you about my favorite scientific passion, the cerebral cortex, particularly in humans, and with the sub-themes that are outlined below. So let's start off with some basics. I hope we'd all agree that a fascinating set of questions relate to how our brains work to make us unique as a species and as individuals with an incredibly diverse range of talents. Unfortunately, many of us are afflicted by devastating brain disorders at various ages and stages of the lifespan. So the challenge is to decipher the brain in health and disease, and that will keep us busy for a century, if not a millennium, should our species survive. So I'm going to focus mainly on healthy brains, but I will touch on issues of brain disorders in the latter parts of the talk. I like numbers about the brain. Let's start with a few fundamentals. So each of us has about 3 pounds worth of squishy tissue inside our skull. It contains about 90 billion neurons. And if we look at a lateral view, we see what's shown on the left here. If we take a slice through the middle portion of the brain, we see an image like that on the right. If we slice it right down the midline and look at it from what we call the medial view, you see what's shown here. And in all three views, the dominant structure is the cerebral cortex, which contains about 80% of brain mass, but remarkably contains only about 20% of the neurons. That's because it's chock-a-block full with more than 100 trillion synapses, 10,000 per individual neuron on average. And on the underlying white matter, there's about 100,000 miles worth of cabling in the white matter to connect different brain regions. The pint-sized cerebellum in the lower left here has only about 10% of brain mass, but it contains 80% of the neurons, and it works together with the cerebral cortex to coordinate our movements and thoughts. The rest of the brain, the subcortical structures, various nuclei that we see, constitute about 8% of brain mass and less than 1% of the total neuronal count, but is incredibly important for various housekeeping functions. I won't have time to talk much about those, but they'll come up now and again. So I want to structure this presentation to talk about various brain basics, comparing brains with computers and genomes very briefly for each. I'll talk about maps and cartography of the cerebral cortex, individual variability and development, and then a segment on cortical connectivity in animal models. Then we'll get to the main theme, the Human Connectome Project and what it's taught us about brain circuitry and also about cortical parcellation that Jerry touched upon already. And then I'll wrap things up. So each neuron, as I said, contains about 10,000 synaptic connections that are made on its dendritic arbors, and it signals its responses to those integrated inputs by firing impulses at a range of anywhere from 1 to 100 per second. So that is embarrassingly slow compared to modern computers, which have chips that operate in clock cycles that are in the gigahertz range. So computers are increasingly powerful. Even Watson could be a chess champion, and that was decades ago. We like to enjoy the illusion that we're still smarter than even the best computers. And the question we want to address in broad strokes is why and how are we smarter? And fundamentally, it has to be in our wiring, our connectomes. A couple of just very general comments to get us going. In computers, memory, storage, and computation in the CPUs are handled by physically separate devices that are closely interlinked but are not the same entities. In brains, the memories are stored in synapses and the computations are done at the level of synaptic integration. So that's a fundamental difference. If we switch over to an analogy, a set of comparisons between brains and genomes, we know that we have a genome sequence that has about 3 billion plus base pairs, and a mouse is nearly at the same, almost 3 billion. We know in terms of the actual number of genes, it's only about 20,000 plus in us and perhaps even a handful more in mice. but they're in the same ballpark. And when we compare homologous genes in the two species, nearly all have homologs, and the experts say that there's about 85% sequence similarity. So that's remarkable. That degree of similarity leads to such divergent results when we use those genetic instructions to generate brains. So a mouse and a human differ in brain size or weight by about a factor of 3,000 and more than a factor of 1,000 in the terms of the number of neurons contained. If we look at the number of anatomical subdivisions we call parcels and loosely analogize those to the number of genes in the genome, we know that there are some hundreds of nuclei in mice and a comparable number, perhaps some additional ones in humans for the subcortical domains. For the cortex, we know, as I'll show you, there are about 40 cortical areas in mice, and in humans, the debate has raged for more than a century as to whether there are Brodmann's estimates of about 50 or so, or another estimate in the ballpark of 200. And I'm going to vote for the latter, but we'll get to that evidence later. If we look at brains of more than just mice and humans, we see that there's this 3,000-fold difference in size between us and mice, and that reflects changes over 75 million years since the divergence of the two lineages. For marmosets, an intensively studied non-human primate, the divergence occurred about 35 million years ago, and the brains are about 200-fold different in size. For the macaque, there's a 15-fold difference in size, and the divergence occurred about 25 million years ago at least. For the chimpanzee, our closest non-human primate relative, there's a 3-fold difference in size, and the divergence occurred about 5 or so million years ago. And in the expansion and leading into the human lineage, most of that expansion occurred in the last 2 million years, so very rapid progression in evolutionary terms. The other thing that's painfully obvious at a glance is that the pattern of cortical convolutions varies tremendously across species, and that is part of what brought me into becoming a cortical cartographer, making maps. So what do we want to make maps of? Well, one of them is what we call cytoarchitecture, the pattern of distribution of cell sizes and cell densities that gives rise to cortical layers, and those patterns, those layering patterns, differ from one region to another. That's what led Brodmann, as Jerry mentioned, to identify upwards of 50, his actual number was 47 areas that he published in 1909. But around the same time and at the same institution in Germany, Cecil and Otto Vogt looked at myelin, which of course is the insulation that wraps axons and can be stained by other methods that turn the white matter jet black, but for our purposes, importantly, lead to patterns of myelinated axons that differ in density and orientation in different cortical regions. And Cecil and Otto Vogt identified about 200 areas. And so this issue of what's the accurate parts list for human cerebral cortex has been hugely challenging to decipher for the past century. The other thing that's obvious from looking at these pictures is that brain drawings, while they're useful and conceptually, are incomplete. They don't show what's buried in various sulci in the cortical folds. And that's true also in Brodmann's map that he published for the Old World monkey. And so when I started working on macaque visual cortex back in 1975, I was struck by the analogy with Earth maps, which of course give us useful two-dimensional flat maps by making a few cuts and flattening the Earth's surface out and using it to represent things like geography, the continents and islands and valleys, and as well as the political subdivisions and tons of other information that can be portrayed on flat maps. So for the macaque, as Jerry touched upon, we made flat maps of macaque cerebral cortex. It was obvious from the outset that this was a job for a computer, but in 1975, they weren't up to this job, so I literally used pencil and tracing paper to make these early maps. And this is a map of what's buried in gray in sulci versus what's exposed on gyri on the outside. And we also use these maps to take information from the literature. So Dan Fellman and I identified from many published studies different cortical areas that could be identified by differences in function, architecture, connectivity, and or topography. And a small number of areas like primary visual cortex V1 and MT were robustly identifiable and gave the same answer by any or all of these methods. But in fact, for most of the rest of the cortical regions in visual cortex shown in color, the boundaries are sharp in the drawing, but the evidence underlying that was very shaky. It was debatable, the boundaries were subtle, different investigators came up with different answers. And so cortical parcellation is a profoundly hard problem, and we need new methods to drill down and get better answers. And one of those sets of answers come from better imaging methods, magnetic resonance imaging, that came into play in the 1990s. So here, for a reminder, is a postmortem slab of real human brain. Here is a matched, roughly T1 weighted, as we call it, structural MRI imaging showing the gray matter and white matter and CSF in dark shades. And since we're going to be using imaging a lot, I'm going to give you, for those of you not intimately familiar with that, a kind of a nano course on MRI technology. So if you go into a scanner, as many of you have, there's an outer core that's the magnet that has a field strength anywhere from 1.5 to 7 Tesla. And then inside that is a concentric ring that generates gradients in that big magnetic field. Inside that, in green, is another ring of electronic elements that generate radio frequency pulses that give rise to the complex things we call pulse sequences. Just around the head is what we call a head coil for brain mapping that detects very faint signals related to these pulse sequences. And then you feed that data into powerful computers that do online reconstruction and then many stages of data analysis that we'll be touching upon. And there are many MRI modalities. The ones we care about for tonight are structural MRI, the T1 weighted you've already seen. You'll see soon an example of T2 weighted that gives a different pattern of structure. Functional MRI that can be related to particular tasks or what we call resting state. And I'll talk a lot about that. Diffusion MRI gives another valuable measure, but I don't have time to talk about it, so we're going to let that one slip by mostly. But collectively, these different approaches and different pulse sequences and imaging modalities give us information about human function, architecture, connectivity, and topography in the living individual. So here's another structural image that's a much higher quality because we use finer grain 0.7 millimeter volume element or voxel sizes. And we use the free surfer method pioneered by Bruce Fischel at MGH to capture the green inner boundary between gray and white matter and the blue outer boundary between white and peel in CSF. Between the peel surface and the CSF. And we capture very accurately the cortical ribbon, including thin and thick regions, and then we model this with a geometric model that we can spin around in the computer, and we can also inflate it to smooth out the wrinkles, but preserve a measure of cortical shape by showing deeper regions buried in sulci as darker on the inflated map. So if we look at another individual, here's a left and right hemisphere cortical reconstruction seen from above, and we can take these and not just inflate them, we can flatten them out like a pancake, now computationally instead of with pencil and tracing paper. And that allows me to remind you that each cerebral hemisphere is about the size of a 13-inch pizza, which for those of you who are thinking about dinner, should make you salivate a little bit. And the cortical areas that we'll be talking more about are on average about the size of an individual pepperoni on that pizza. If we look again at the view from above, you can see that the convolutions are similar but far from identical in the left and right hemispheres. So for example, these arrows show very different patterns in the parietal lobe. And if we look more systematically across individuals instead of just between left and right hemispheres, this individual variability comes out very prominently as well. So here are 4 healthy young adults from a lateral view of the right hemisphere. They're different, but it's hard to appreciate some of the nuances, so we get a better view by making these inflated maps and maps of what we call sulcal depth. And you can see that everyone has a consistently arranged central sulcus on the inflated map, but in many other regions, such as the boundary between the occipital and temporal cortex, the folding pattern is dramatically different from one individual to another. Remarkably, this difference is true even for individuals with the same genetic instructions, that is, identical twins. So here's one twin pair, and look how different their folding patterns are. Here's another twin pair. So there is some heritability to the folding patterns, but that's dominated by the individual variability. Another important basic fact is that the cortical areas that we've touched upon in monkeys, and we'll say more about in humans, but just to get one point out there, if one identifies primary visual cortex V1, which can be done very accurately, the individual to individual variability is more than twofold. So we don't have identical maps, and moreover, the folding pattern is not consistently related to the areal boundaries in many regions. There's some correlation, but there's a lot of variability.
What's the function of the folding pattern?
Stay tuned. Excellent question. What makes the cortex fold and what does it signify? Well, let's first take into the up for brief consideration when folding occurs. So here is a healthy individual infant this was a preterm infant that was imaged right at the beginning of the third trimester, between about 6 months of gestation. And you can see in the coronal slice and on the surface reconstruction that the folding pattern is very incomplete. It's very smooth. And yet, several months later, just at the time of birth, in this individual, the folding pattern is nearly as complex as it is in an adult, even though the brain has threefold more growth yet to occur. And we'll come back to that later in the talk. But the fact that the folding occurs mostly prenatally is something that I started thinking about when Tom Coogan and I looked at this issue in macaque monkeys. And what we found was that in the posterior regions between V1 and V2, the connections that we anatomically assayed, the connections between V1 and V2 going in both directions, developed by projections of axons through the white matter, and they developed right around the time that folding occurs, forming a gyral crease or crown in between V1 and V2. And one night while sitting in my living room couch, a light bulb moment came to pass, which was that maybe this was not accidental, maybe there's a causal relationship, and in particular, suppose that mechanical tension along the axons connecting one visual area or one cortical area with another, suppose that mechanical tension pulled strongly connected regions together, forming gyral regions in between, and in the opposite direction where a sulcus forms, the hypothesis is that there aren't enough connections to fight against the tide and to allow a sulcal fold to occur. So this one hypothesis, mechanical tension drives cortical folding, can account for consistency in strongly connected regions and variability in regions that are essentially balkanized little areas that may have very different sizes and connectivity patterns. So this was 2 decades ago and still is a very attractive hypothesis. It's definitely not proven, it's actually controversial, but I think it's very likely to be one of several mechanisms along with differential proliferation and some kind of mechanical buckling. So I won't try to debate the merits of that any further, but I want to draw attention to another intriguing issue that has received less attention, but I think is equally important. Not only is the cortex folded, but it remains a thin sheet of variable thickness across its entire extent. So why is the cortex a sheet-like structure and not a big blob-like subcortical structures and nuclei? And my argument is that it's related to the anisotropies of dendritic arbors, particularly, and in cortex, the dominant anisotropy relates to the radial orientation of pyramidal cell dendrites. And all we have to do is invoke the hypothesis that mechanical tension along dendrites as well as axons that I hypothesized previously, tension along dendrites would bring superficial and deep layers close together and letting cortical expansion occur mainly by tangential expansion, not radial. And so that's an attractive hypothesis, again, but not proven. So cortical folding is, we have a mechanistic hypothesis for them, and it's worth noting that abnormalities in cortical folding occur in a number of brain disorders. So we first reported on that in a particular situation of Williams syndrome, but there's also an example I'll illustrate for folding patterns that are abnormal in preterm birth. So here are healthy term infants, similar to the example you saw a few moments ago. The folding pattern is very mature looking. But if you take individuals who were born prematurely around the end of the beginning of the third trimester and look at their folding patterns at term equivalent, the same age as you see on the left, you can see that the folding pattern is very abnormal, immature looking, particularly in lateral temporal cortex, also in prefrontal cortex. So there's maybe a selective vulnerability in these regions, and the proposition is that improved imaging of structure and function may aid in diagnosis and treatment of the failure during the growth in the neonatal intensive care unit to form proper cortical folding. So our last brain basics coverage relates to cortical connectivity. And I first want to introduce the idea of a connectome. So a genome we've already covered is the complete sequence of base pairs in an individual genome. A connectome concept is a comprehensive map of neuronal connections. But we absolutely must put this in quotes, what we mean by comprehensive, because it's impossible to do total reconstructions of everything. So there instead is a world of very exciting progress in what we call the microconnectome domain, using electron microscopy and other ultra-high resolution methods to reconstruct every axon, dendrite, synapse, cell body, glial cell, and identify their entire extent, but only in a region that has yet to exceed a cubic millimeter in total volume. Then there's a mesoconnectome world in which one can look at local and long distance connections at very high resolution, but not ultrastructural yet. But that has to be done by tracer injections or other invasive methods in many different animal model individuals. I'll be talking mainly about the macroconnectome world, which uses in vivo imaging mainly and uses volume elements or voxels that are 1 to 2 millimeters on a side to capture information about brain structure, function, and connectivity. So that's what the HCP project we'll talk about in a short while covers. But I want to introduce some of the important constraints in our understanding of brain circuitry by looking at mesoconnectome style analyses in animal models, starting with the mouse. So here's a mouse brain from above. Here's our schematic diagram of what axonal dendritic and axonal arbors look like. Here's the reality coming up on the right here. So there are powerful methods now available for reconstructing in entirety the dendrites and the elaborate axonal processes in individual mouse neurons. So here's one from an Economo et al. Study. And here's another example coming up from the Allen Brain Institute. So this is another mouse cortical neuron. And if we look at it in 3D, you can see the richness of axonal arbors locally, and then a single axon extending down into subcortical structures, jumping across to the other hemisphere, and look at the complexity of branching patterns that we aspire to understand in brain circuitry. If we relate that concept of the richness of axonal connectivity to brain parcels in the mouse, Andreas Burkhalter at WashU has pioneered a method of making flat maps of mouse cortex, not computationally, but literally by taking the mouse brain lightly fixated and flattening it out like a pancake between two glass slides, and then slicing tangentially and showing that one can use various methods, architectonic and connectivity measures, to make maps of the mouse cerebral cortex that identify, in the latest version, 41 distinct cortical areas. So that's how we get that ballpark estimate. And if we look at the connectivity in relation to those cortical parcellations, we can put retrograde tracers, say, into V1, and those tracers are picked up at synapses and taken retrogradely back to the cell bodies. And every little yellow dot here is an individual cell body whose axon projects to V1. And you can see there's a lot of yellow in many regions, but if we look at very closely and quantitatively, even in some regions like somatosensory cortex, there's a handful of connections showing some degree of connectivity, and we can turn that into a connectivity matrix in which all of the connections of V1 are shown in this vertical column, and all of the connections of 19 areas in total are mapped in this parcellated connectivity matrix, which shows some degree of colorations indicating connectivity strengths varying over literally 5 orders of magnitude from the weakest to the strongest in this connectivity matrix. So that's a general principle of rich distributed connectivity that we're learning about from mice. If we do the same kinds of experiments from the Kennedy lab in the macaque monkey, you see again very rich interconnectivity. About 60% of all possible connections are actually found to be present, but the same richness of connectivity strengths. And ditto for the marmoset from the Rosa group in Australia. So these general principles are the current lay of the land for our understanding of cortical circuitry. So that contrasts with the schematic diagram of the wiring of the macaque visual cortex that Dan Fellman and I published 20 years ago, where we identified 10 levels of a distributed hierarchy, but we showed the wires as just connections present or absent, and we showed the connectivity matrix as a binary matrix with hundreds of pathways. Now we know from this repeat of the macaque weighted connectome that there are many more pathways, thousands of pathways when the full matrix is done, and the tremendous range of connection strength. So we've come a long way. We've got a long ways to go. We need better parcellations than the one that's shown here. This is not a completely accurate map. And we'd also like to get maps of dense connectivity that are free of any particular parcellation assignment. And we'd also like to know how this generalizes to human cerebral cortex, and that lets us shift now to the human connectome project that has been touched upon before. So I first want to introduce the major modalities and then talk briefly about the relationship between functional and anatomical connectivity. So the Human Connectome Project was funded from 2010 to 2016, and it acquired data on brain structure, function, and connectivity from healthy adults, twins and their non-twin siblings, actually slightly more females than males for the record, using improved scanners and pulse sequences, and an unprecedented 4 hours of imaging data from each of these 1,100 subjects that we were able to scan. And in addition, we got extensive behavioral and demographic data. Genotyping was done, that's now available. And we put a lot of effort into analyzing the data using improved analysis. We call them pre-processing stream pipelines that gave better alignment across individuals and better visualization, as well as connectivity and behavioral analysis and parcellations that I'll focus on in a moment. We also, as Jerry touched upon, put a great effort into sharing the data in a user-friendly way for the data, imaging data itself, as well as the methods and tools that we've touched upon. And more than 10,000 investigators have tapped into these data sets, and the number of publications grows rapidly week by week, but is well over 600 at this stage. This point about using better methods, we've articulated in a systematic way into what we call the HTP style neuroimaging paradigm that differs from traditional neuroimaging in its 7 core tenets. So the idea is the 7 bullet points I'll go quickly through: get as much multimodal data as possible from each individual and lots of individuals in a study. get it at maximal resolution in space and time, as well as the highest data quality possible. Avoid distortions and blurring of data that has been tolerated in traditional studies but need not be the case anymore. Respect the geometry of brain structures, and in particular use cortical surface models for analyzing information related to cerebral cortex. Align the data precisely across individuals, which can now be done much more accurately than used to be the case. And analyze the data using an accurate brain parcellation that I'll touch upon in a short while. And then freely share the data, not only the raw and the minimally preprocessed data, but the publication-related data in ways that I'll show in a moment. And for those of you who are interested or know colleagues who might be interested in learning more about what's under the hood, we have an annual course. This year's HTP course will be in Portland, July 8 to 12. And if anyone's interested, they can just Google and tap into that. We encourage participation. What do we get for our money? Well, first of all, I'll remind you that in terms of looking at cortical structure, we have as a classical approach postmortem analyses of cytoarchitecture on the left, myeloarchitecture on the right. Can we get information relating to these from in vivo neuroimaging? And the answer is yes. And my engagement with this started when an exceptionally talented student, Matt Glasser, came to my lab in 2008. And he had noticed even before arrival that in conventional T1 weighted images, this is now a horizontal slice, that in the cortical gray matter ribbon, some regions are a little brighter than average, other regions are a little darker than average. And then if you take a T2 weighted image, different pulse sequences giving a different pattern that's essentially the mirror image in contrast, in the gray matter and a T2 weighted image, the patches that had been brighter are now darker than average. The ones that had been darker are now brighter than average. So Matt cleverly did the math, took the ratio of T1 versus T2 weighted values for each voxel or volume element, colorized it to make it more attractive, and then mapped it accurately to the cortical surface to generate maps that turn out to be strikingly well related to maps of myelin content from postmortem analyses. And so we call these myelin maps, and the red represents heavily myelinated regions, the blue and indigo represent lightly myelinated regions, and other colors in between. If we do this for hundreds of subjects and align it very accurately in ways that I don't have time to detail but are very important, we get very crisp-looking myelin maps on the left here. And if we look at the cortical thickness using the free surfer models that I mentioned before, we get thickness maps on the right. And in both cases, we can see features of systematic variations in myelin content or thickness that are strongly related to areal boundaries. We call these areal features, and we use spatial gradients, what we call gradient ridges, to look for evidence for areal boundaries, which are very informative for some regions, but in prefrontal and other regions, the lightly myelinated regions that we are associating with cognitive processing regions, the structural measures of T1 and T2 aren't so informative, so we draw on other modalities, particularly functional MRI. So for that, Deanna Bartsch spearheaded the efforts to get high-quality task MRI data, and we got an hour's worth of data from each participant using 7 different tasks run in sequence. I'm going to quickly illustrate 3 of them. One of them is a language task, actually listening to stories in the scanner versus doing arithmetic in the scanner. Another is what we call a social cognition task, so looking at these simple images and asking, are these images, these triangles, just moving randomly past one another, or are they interacting in a social-ish way, hugging and kissing and dancing in ways that make a big difference inside the brain? And the third exemplar task is what we call working memory. So is the image one is looking at right now the same or different from the one two previously, a so-called two-back process where we had different types of images to help us in analyzing the data. So we can look at this task fMRI data mapped onto cortical surface models that are inflated, as you see here on the left, on subcortical slices, and also on a cerebellar flat map shown in the lower right. So just to give you a very quick glimpse at what happens inside your brain or HCP subject's brain during these kinds of tasks, when listening to stories versus the baseline contrast, that lights up in yellow and red patches of cortex, swaths of cortex in lateral temporal, prefrontal regions in different patches in both the left and right hemispheres, although it's stronger on the left as expected. If we take the next task, the social versus random, we see a strikingly different pattern. There's some overlap, but mostly different regions, and the quality of the alignment lets us separate regions that would have been blurred by conventional analysis and visualization methods. And the third task is the working memory, which lights up yet a different region or set of regions, particularly in lateral parietal and prefrontal cortex. So just to summarize quickly, each task that one does in a scanner engages a complex network of brain regions that overlap to some degree. They're typically not totally isolated, but they're rich in information of spatial organization. If we go to a different modality, again using functional MRI, but now using what we call resting state, lying quietly in the scanner, staying awake and thinking about what's for dinner, who's going to win the basketball playoffs, etc. That leads to a complex symphony of fluctuations in space and time across different brain regions. And one can analyze that by taking any pair of gray matter locations in the cortical or subcortical regions, looking at their functional MRI time course after cleaning out noise that complicates the issue, and ask, are the patterns correlated perfectly or imperfectly, or are they even anti-correlated? And that leads to all pairwise comparisons of correlation, giving us what we call a dense functional connectivity matrix, or a dense connectome, shown by this matrix over here. And we can sample that one location at a time, take one region, for instance, in lateral parietal cortex on the right hemisphere, and ask who's correlated with that along that row. And one sees a functional connectivity map for this exemplar seed in parietal cortex, a totally different pattern from a central sulcus pattern. And this is very informative and has led to thousands of studies around the scientific literature, but it raises the fundamentally important question: to what extent do these functional correlations that we label as functional connectivity, how much do they actually relate to anatomical connectivity? That's currently essentially impossible to address incisively in humans, but we can turn our attention to animal models. So why do we care about this? Brain circuitry mediates behavior, as I already argued. For human cortex and brain, our ability to infer connectivity is related to two different modalities: functional connectivity that I've introduced and we'll talk more about. I'll just mention that what we call structural connectivity uses a completely different modality, diffusion imaging, and an analysis that we call tractography to infer estimates of connectivity. But both of these methods are highly indirect. They have serious limitations that are progressively being better appreciated. But we care about this not only because of our fascination about brain circuits, but also because many brain disorders are hypothesized to be abnormalities in connectivity, a so-called connectopathy. And if we want to get a better handle on this, we need better analysis, critical assessment of whether these methods actually yield robust insights about real brain circuits. And to do that, we really need animal models. So that's part of why I introduced the macaque and marmoset animal models as substrates for getting accurate anatomical connectivity data. So to bring us back into macaque connectivity, this is an example of a retrograde tracer injected into one brain region, and all of the retrograde labeled cells are shown on this for this particular slice in little patches of gray. If we take this slice and just look at the contours, we can identify on our MRI-based macaque atlas a corresponding contour. And we can do that for every slice that has tracer data in it, and use that strategy to map the distribution of connectivity from a single tracer injection, in this case in ventromedial prefrontal cortex, throughout the entire hemisphere, whether it's weak or strong. And we can do that for another injection in a different brain region, still another injection, and so far we've done that on 28 different example injections, but we're continuing to do this work with Henry Kennedy's lab to get accurate maps of real connectivity. Then in a parallel collaboration with Takuya Hayashi's lab in Japan, we're looking at functional connectivity from a different set of macaque monkeys, but using high quality data acquisition and analysis, Takuya has been able to analyze 30 different anesthetized macaques and to generate analyses of seed-based functional connectivity using resting state fMRI. So here's a seed in one region of medial prefrontal cortex, and you can see a pattern of functional connectivity on the inflated map and on a flat map. And if we move the seed location a little bit, you see a totally different pattern showing that this method is informative, but we can now compare anatomical and functional connectivity using matched seeds. So here is an anatomical tracer injection in a premotor area F4, and here's the functional connectivity, which shows many similarities but many differences. And a different seed location in prefrontal cortex shows a different anatomical connectivity, and in the middle panel here, a different functional connectivity. They are correlated but far from perfectly. And if we do this quantitatively and ask what is the correlation between each tracer injection anatomical connectivity and functional connectivity, we get values where the correlation using what we call full correlation, the most straightforward analysis, is up to 0.6 or more, so not fantastic, but pretty good, and it falls off for different areas. And one can do different analysis methods, one called partial correlation that I'll not try to explain in detail, and get different estimates. So the point at this stage of the exercise is to say we're making headway in quantifying the degree to which anatomical and functional connectivity relate to one another, but we have a long ways to go, but we're getting more data and have different analysis strategies to bring to bear. So with that as background, let's now move on to human cortical parcellation, which I've introduced previously by showing you at the very beginning Brodmann's cytoarchitectonic map, the Vogt's much more fine-grained myeloarchitectonic map. In recent years, a number of investigators have used fMRI, particularly resting state fMRI that we've already talked about, to estimate cortical organization and to parcellate the cerebral cortex, as shown here for Randy Buckner's and Thomas Yeo's parcellation as one example. Evan Gordon and Steve Peterson's lab came up with a different parcellation that has more than 400 parcels, but it has issues with it. One is that the map is incomplete. There are some regions that weren't accurately modeled at all by functional MRI. And I think perhaps more concerning, the maps, while rich on both hemispheres, showed many asymmetries that are not convincingly neurobiologically robust, I would say. So in general, these examples and many other efforts to parcellate the cerebral cortex using a single modality, be it architectonics on its own or fMRI on its own, are underpowered for given the challenges at hand. So Matt Glasser and I aim to get better parcellations using a multimodal HCP approach that uses all four types of information hand and glove, and as well to actually be able to parcellate individual subjects as well as group averages. So I'm going to show you the strategy we use very briefly. So here's a myelin map that you've already seen, and here is the spatial gradient in the myelin map. And we're going to key on one particular region of interest. This white outline represents the regions of a sharp gradient in myelin content, and we label it 55B as a marker for reasons that will become apparent in just a moment. But next we note that the task MRI contrast, the language versus, or story versus baseline, lights up this little patch inside the 55B outline, and the spatial gradient shows it is completely surrounded by this gradient ridge. If we use resting state fMRI and put a seed location in one location, it shows strong functional connectivity with the region inside the box. And if we take a different seed, it lights up other regions that are related to eye movements. And the spatial gradient by this third modality agrees with the task fMRI and the myelin gradients. So all three modalities essentially give the same answer, and that's critically important to our approach. Matt and I call this area 55B because a half century ago, Adolf Hopf had identified a lightly myelinated region in what we consider to be pretty much the same point, and so we had to give it a tag. We chose history rather than completely novel naming scheme. And Matt was able to apply this general gradient-based multimodal approach to all of cerebral cortex in both hemispheres. And the result is this multimodal parcellation shown here, in which we identified 180 areas in the left hemisphere, a matching set of not perfect but fairly symmetric, same 180 areas on the right hemisphere. They're colorized here according to the functional association related to functional connectivity to identify visual areas in bluish hues, sensory motor areas in green, auditory in red, regions that are activated during tasks in bright hues, regions that are turned off during tasks and more strong in the resting state, the so-called task negative in dark. And we also gave these areas labels, some of them for regions that matched what's in the published literature from dozens of different studies. We were able to use the same published name for 83 different cortical areas, but 97 of the 180 areas we essentially gave new names, although in many cases it was splitting, for instance, TE1P versus TE1M and TE1A according to their finer grained parcellation of a published ARC parcellation. So this is a group average parcellation. As I touched upon a moment ago, Matt was able to introduce what we call an areal classifier algorithm to identify each of these 180 areas in nearly all individuals. And I'll just show you an example of that in a moment. But I want to comment briefly on the data sharing aspects of this, which, as Jerry alluded to, has been near and dear to me. So we were able to generate a publication with many different figures using our Connectome Workbench software, and we take the data as generated by the software, generate fully annotated images, publish the images, and then we upload the data associated with that image to our database that we call BALSA, and at each of the figure legends in the published online article, you just click on that URL, and that takes you immediately to our BALSA database, and in particular to the access point for the particular data set we call a scene file that lets you say, here's the image on a preview postage stamp size in the database. If you actually want it, you log in, click on a download option, and in short order, you've downloaded to your laptop or computer at home the data set that lets you launch the visualization and see exactly what was in the paper and navigate it, rotate it around, or manipulate the data and make good use of it for whatever purposes you want. And you can get, for example, downloaded labeled parcellations if you want to try to better remember who's called what in this parcellation. The ability to take not only a group average that you see here, but individual subject parcellations I mentioned before, and that takes advantage of an aerial classifier method whose exact operation I don't have time to spell out, but basically it works very well. Here's a parcellation in white boundaries overlaid on an individual subject's myelin map for one individual. Here's one particular area. The same area in a different individual has a somewhat different size, shape, and exact location, but it's identified robustly by the classifier, and it confirms our expectations that areas vary in size, shape, and precise location, but we have the data now to enable a variety of follow-up analyses. We can also take this data set and analyze resting state networks. So this is a study done by our colleagues at Yale and Princeton, the so-called Cole-Anticevich network partition. And so basically each of the colors here represents, for example, in yellow, a group of areas that have similar functional connectivity and are strongly connected with one another and are organized into these resting state networks, of which there are 12 in this particular scheme. And this is the functional connectivity matrix that allowed Cole and Anticevich to identify these networks, and they are organized into networks related to sensory, vision, audition, and so on, language, and other cognitive functions. And it's not shown here, but this analysis also gave the very useful parcellation of subcortical structures, hundreds of subcortical ones, as well as the cortical maps. So this is a very rich addendum to the MATS multimodal parcellation. So in the remaining moments, I want to touch upon two distinct themes: evolution and clinical issues, brain disorders. So each one will be relatively brief, but I hope you find interesting. Here is the human myelin map that you've already seen for reproduction of the human pattern. Here in the middle panel is the myelin map for the chimpanzee. Next over is the macaque myelin map, and then the tiny marmoset's myelin map. So in each case, we can identify regions that are very likely to be homologous. The MT, motion processing complex, is one of the more striking. And so this gives us access to inferences about evolutionary homologies. The lightly myelinated regions that are present in all species are at a glance much more prominent in humans compared to the non-human primates, and are particularly small over in the marmoset version. We can look at these evolutionary issues in the context of other approaches. So if we take our myelin maps for human and macaque and drill down on them and compare them by making a mapping between macaque and human that draws upon a set of colored regions here that are regions, cortical areas that are likely to be homologous across species. So this is a mapping between monkey and human cortex that allows us to estimate the degree of expansion associated with these different evolutionary lineages. So this is what we call a cortical expansion map in human versus macaque. And you can see the reddish regions are the regions of maximal evolutionary expansion in lateral, parietal, temporal, and prefrontal cortex that is dramatically greater than the very modest expansion in early sensory areas especially. This is a replication of this expansion map related to evolution that we're going to now put side by side with what we call a developmental expansion map. So remember, I told you briefly a little while ago that at term equivalent, the cortex is about 1/3 the surface area of the adult form. And if we look at a relationship between these two, we see that developmental expansion postnatally is not uniform, and the hotspots of developmental expansion are more modest quantitatively, but strikingly they agree in rough terms with the hotspots of evolutionary expansion. So regions that are associated with higher cognition are evolutionarily expanded. They expand preferentially in postnatal development. They are the lightly myelinated regions that you've already seen. And one last addendum that I think is quite nice, if we take advantage of the cellular level analyses of Guy Elston and his colleagues, they've shown that with selected sampling of early sensory areas, cell bodies and dendritic arbors are small in densely myelinated regions. In higher cognitive regions like TE and prefrontal cortex, the dendritic arbors are much larger and there are more synaptic density per cell. And so the idea is that regions of low myelin and have large dendrites, more synapses, and may well be associated with greater synaptic plasticity, although that's obviously speculative. Shifting over to our last major topic, brain disorders. Just in broad strokes, everyone either has a disorder or knows somebody with one. That's just the unfortunate reality. It has enormous social and economic impact. Brains malfunction in an incredibly diverse number of ways. We're far from having cures, and treatments that we have are limited efficacy in most cases. We don't understand what's gone wrong, and what we need to advance the frontiers are a combination of better basic neuroscience in humans and animal models, greater acceleration of technical advances such as those that have already made progress with the BRAIN initiative, better translational neuroscience, improved diagnosis and treatment, and I'm going to give just two quick examples in the remaining moments. So one is clinical depression, which is all too common. One way of treating severe depression is with deep brain stimulation of the subgenual prefrontal cortex, roughly here. Another way that's emerged that has some potential but is still early days is transcranial magnetic stimulation, putting a magnetic stimulating pulse above the head in various locations. And it turns out that works well in some individuals and not so well in others. And a recent study by Drysdale et al. Has shown that diversity in functional or clinical responsiveness is related to brain circuitry as inferred from resting state functional connectivity analyses. And the hope would be that better, more careful analyses of resting state functional networks in individual subjects may improve the specificity with which we both stimulate appropriate regions and interpret the data. So that's a way forward for the future. Jump over to autism, which is obviously of high interest to the Simons Foundation. Some studies report what they call hyperconnectivity using functional MRI, greater inferred connectivity. Others report weakened connectivity, hypoconnectivity of particular networks. A very recent study from Udin's group reported no differences between functional connectivity across comparing with autism, typical development, and ADHD. And they interpreted this as a likely more plausible interpretation relative to the false negatives and false positives reported in earlier studies. But what I want to draw attention to is that the methods used in most of these studies are what I would call the traditional paradigm that I've already critiqued. They are based on rather short scanning data, non-optimal analysis methods, and the HCP style paradigm that I've already introduced, I claim, and with considerable evidence to support it, is fundamentally more sensitive and a better approach. So I'm not going to walk through these again, but just remind you there are better ways of going about business. And the hope is that doing this in healthy individuals, such as the lifespan projects in aging and development that are currently underway, the adolescent brain and cognitive development is a parallel study that's getting over 10,000 children that will be followed for a decade. There's a baby connectome project and a developing human connectome project that are using cutting edge methods. There are disease connectome projects that are currently underway that will look at a number of disorders. Unfortunately, autism is not yet one of them, but hopefully something pushing the envelope on autism will also emerge. To wrap it up, what are the most important bottom lines? Cortical parcellation is fundamental for understanding brain organization. We have evidence for 180 areas per hemisphere in human, smaller numbers in animal models. Cortical connectivity shows a very complex distributed hierarchical organization with a tremendous range of connection strengths. We're far from having a human connectome that spans the macro through the meso and micro level. Evolution has given us evidence for preferential expansion, particularly associated with higher cognitive regions. And HCP-style studies of disease development and aging are critical for understanding and improving diagnosis and treatment. So maps are key to understanding for both Earth and brains. Better maps make for better analyses, all the way up to satellite imagery, structural and functional MRI. Google Earth has been transformative for our understanding of navigating the Earth, and connectomics and the associated methodology improvements will hopefully accelerate progress in studies of brains. Everything I said relied tremendously from contributions of literally hundreds of individuals, HCP Project and more. I highlight a few names, Matt Glasser in particular, Deanna Barch, Camille, Steve, and Dan, and people in my lab have also contributed greatly. Funding has been crucial, and the clock has run out, and I hope there's time for a little bit of discussion and questions.